Most of the food we eat is ultimately derived from crops grown on farms. For example, the bread we eat is derived from wheat crops, the margarine from oilseed rape crops, and strawberries are harvested directly from farms. However, our food security is threatened by crop diseases, which may cause loss in yield or may make the produce unfit for human consumption. It is estimated that globally crop diseases cause a 15% loss in yields of crops, with the problems being most acute for subsistence farmers who have few options in terms of disease control. It has also been estimated that if we could save 1% of the losses from crop diseases, another 25 million people in the world would have enough to eat. So, could you tell me a little bit about some of the measures that you use to control disease?
For foamer and light leaf spot, we're spraying in the autumn and sometimes also again in the spring.
Light leaf spot and foamer we tend not to have problems with because mainly I choose varieties with very good resistance ratings.
And it's resistance that this paper, published in Trends in Plant Science, particularly relates to.
Like in animals, the plant immune system has to contend with intra- and extracellular pathogens. Our findings enhance the traditional understanding of the plant defense system and introduce a new concept explaining how plants protect themselves against pathogens that grow in the space outside plant cells, the apoplast. We propose that this new concept is called Effector-triggered defense, or ETD. Plants and animals share a common mechanism of initial pathogen recognition. The innate immune system of humans uses toll-like receptors to detect microbial patterns. Similar surface proteins are used to protect plants against non-adaptive pathogens. This defense response is referred to as pattern-triggered immunity, or PTI. Adapted pathogens can overcome this first line of defense by secreting effectors that manipulate the host response. We propose that the recognition of microbial effectors differs between extracellular and cell-penetrating or haustoria-forming pathogens. Unlike haustorial pathogens, which are recognized by intracellular receptors, effectors of apoplastic pathogens are detected at the cell surface by receptor-like proteins, or RLPs. The signaling pathways and defense responses also differ in response to these different pathogens. Knowing that extracellular pathogens are controlled by RLPs has important implications for resistance breeding.
Different plant pathogens operate differently in their hosts. We're interested in extracellular pathogens that grow outside host cells. They are fundamentally different from intracellular pathogens that form haustoria like flax rust and penetrate host cells soon after invasion. I'm working with Leptosphaeria maculans, which is a pathogen causing foama stem canker on oilseed rape. When we have a susceptible cultivar or a pathogen race expressing the Avernal allele of the gene, there is a typical disease symptom development. One of the R genes in oilseed rape that recognizes Leptosphaeria maculans is the gene LEPR3. This gene encodes a receptor-like protein, or RLP.
This work is relevant to a wide range of crop pathogens that colonize their crop hosts by growing outside the cell. We propose that breeders should search for resistance genes that code for RLPs in trying to develop improved resistance against such pathogens. And this should help breeders and the rest of our industry in developing more effective resistance against these pathogens, and ultimately will help us in the battle for global food security to feed the world's growing population.